Imagine a high-stakes molecular heist.
Oh, I like where this is going.
Right, so you have this highly effective security system, like a targeted cancer drug, and it has completely cornered a group of rogue cells. The vault is locked down, the perimeter is secured, and, well, the cancer is dying.
As it should be.
Exactly. But then, seemingly out of nowhere, these targeted cells they survive. And they don't do it by breaking the locks or, overpowering the guards. Instead, they completely change their diet. They basically tunnel out of the vault by gorging on a totally different fuel source.
It's a brilliant, if terrifying, evolutionary workaround. You apply this massive lethal pressure, and the biological system just rewires its own internal economy to stay alive.
Yeah, and that exact heist is happening right now in high-grade ovarian cancer. So our mission today, welcome to the deep dive, by the way, is to unpack a fascinating 2026 study from Cell Death and Disease by Hahn and colleagues. They actually caught these rogue cells in the act.
You really did.
They figured out how these cells dodge our best drugs, and more importantly, how cutting off this very specific cellular fuel supply might be the key to beating them. Okay, let's unpack this. Because understanding this process isn't just about oncology for you listening. It's really a masterclass in how biological systems adapt under pressure.
Right, which applies to everything from, ecology to just everyday problem solving. But here we're looking at it through the lens of PARP inhibitors.
Which are a huge deal in cancer treatment.
Absolutely. So high-grade serous ovarian cancer, or HGSOC, it's the most lethal gynecological malignancy, mostly because of late-stage diagnosis.
And it comes back, right? Frequent recurrence.
Exactly. But drugs like olaparib, these PRP inhibitors, have been revolutionary, especially for patients with specific DNA repair defects, like BRCA mutations. They essentially weaponize the cancer's own broken machinery.
Right. It's that concept of synthetic lethality. You use the drug to knock out their backup DNA repair, and since their primary repair is already broken, the cell just accumulates fatal damage and dies.
It's very elegant. But the clinical reality is this massive hurdle of acquired resistance. Over time, the tumors they figure out a way to survive the olaparib. And for a long time, how they did it was just a total black box.
So how did the researchers crack the box?
They used something called non-targeted metabolomic profiling.
Non-targeted meaning they didn't know what they were looking for.
Exactly. They didn't bias the search. They took standard ovarian cancer cells and compared them to a line of scola cells, which are cells they had exposed to olaparib for so long that they became resistant.
Got it.
And by keeping it non-targeted, they just took a snapshot of the entire metabolic economy of the cell. Thousands of metabolites. And one specific pathway was just drastically upregulated in the resistant cells.
And that was glutamine, right? The resistant cells were completely addicted to glutamine.
Oh, absolutely devouring it.
Yeah.
Which, biochemically, makes sense. Glutamine isn't just a basic building block for proteins. It's incredibly versatile.
How so?
Well, it fuels anaplerosis.
Okay, big word. Anaplerosis.
Right. Basically, it replenishes the carbon intermediates of the TCA cycle. It keeps the cellular energy engine running. Plus, it provides the nitrogen precursors for making nucleotides, which you need to build DNA, and it actively turns on pro-growth signals.
Okay, but wait. Cells can make their own glutamine from scratch, right? Like de novo synthesis. So why is this a vulnerability? If they need it, why not just make it internally?
That comes down to thermodynamics and just the sheer scale of the demand. Yes, they have glutamine synthetase to make it. But a drug-resistant cancer cell is a system running in absolute overdrive.
It's panicking.
It is panicking. It's trying to repair shattered DNA and stay alive. So the internal manufacturing process just cannot keep up. The demand vastly outpaces what they can make. They have to import it.
Okay, so think of the cancer cell as a factory running in overdrive. It can't manufacture its own raw materials fast enough, so it has to build extra loading docks on the outside to pull in massive shipments of glutamine.
I love that analogy, yes. And the main loading dock for this job is a protein called SLC1A5. It's a highly specific glutamine transporter on the cell surface.
And the study found this was upregulated.
Significantly.
Yeah.
And not just in a plastic petri dish. They looked at human clinical samples. And this SLC1A5 protein is highly expressed in actual HGSOC tissues compared to normal fallopian tubes.
Oh, wow. So real patients.
Yeah. And having high levels of these loading docks directly correlates with poor patient survival.
So this is a huge deal. But here's where it gets really interesting for me. When the researchers treated the cells with olaparib, the physical amount of the SLC1A5 protein went way up.
Right.
But when they measured the mRNA transcripts, the blueprints for the protein, those levels didn't change at all, so they aren't printing more blueprints.
No, they aren't. And that disconnect between the mRNA and the protein tells us the regulation is happening post-translationally.
Meaning after the protein is already built.
Exactly. The cell is just stopping the existing proteins from being thrown into the cellular trash.
Okay, so how does a cell normally take out the trash?
Through ubiquitination. When a protein is done or damaged, enzymes slap these ubiquitin molecules onto it. Specifically, it's called a K48-linked polyubiquitin chain.
Like a molecular trash tag.
A trash tag, perfect. That tag directs the protein to the 26S proteasome, which is the cell's garbage disposal. It shreds the protein down.
So the cancer cell is somehow dodging this. It's keeping the loading docks open by ripping off the trash tags.
Yes, and to find the culprit doing the ripping, the researchers use mass spectrometry. They basically used the SLC1A5 dock as bait, pulled it out, and sequenced everything attached to it. They found 621 interacting proteins.
621. How do you even narrow that down?
They used molecular docking simulations. And they found one clear culprit, USP14. It's a dubiquitinase, or a DUB.
So it undoes the ubiquitination.
Right. It has a specific region called the UBL domain that docks directly onto the SLC1A5 transporter. Once it's docked, it acts like chemical scissors and just snips those K48 trash tags right off.
Okay, so if the K48 is the trash tag, USP14 is like this dedicated bodyguard. It stands by the loading dock, and every time the cell tries to tag the dock for the garbage, the bodyguard rips the tag off.
Exactly.
And the dock stays open. That is wild.
And it gets wilder. The researchers found that treating the cells with olaparib actually strengthens that bond between the bodyguard and the dock.
Wait, really?
Yeah. The drug meant to kill the cell inadvertently triggers a shift that clamps the USP14 scissors tighter to the transporter. It rescues it from degradation.
So the drug triggers the exact defense system that defeats it. Unbelievable. Okay, we are about 5 minutes in. Let's do a quick mini recap for you listening to just lock this knowledge in.
Good idea.
Okay, bullet 1. Ovarian cancer cells resist these targeted drugs by gorging on glutamine. Bullet 2, they pull all this glutamine in by building extra SLC1A5 loading docks. And bullet 3, the drug itself accidentally triggers the USP14 bodyguard to rip the trash tags off those docks, keeping them permanently open.
That's the perfect summary.
So what stands out to you so far? For me, it's definitely that paradoxical effect, how the drug accidentally trains the cancer's defense. It's just crazy.
It really is the ultimate biological irony.
Okay, moving into the next phase of the heist. Now that the loading docks are wedged open and glutamine is just flooding in, what does the cell actually do with it? Like, how does glutamine stop olaparib from shattering the DNA?
It fuels a massive biochemical chain reaction. First, it goes into synthesizing glutathione, or GSH.
Okay, what does GSH do?
GSH is a chemical sponge. It soaks up and neutralizes reactive oxygen species, or ROS.
And ROS are like toxic byproducts of the drug stress.
Exactly. High doses of PARP inhibitors create severe oxidative stress, superoxide radicals, hydrogen peroxide stuff that damages the cell and triggers death. But by hoarding glutamine and making tons of GSH, the cancer cell just sponges it all up.
So it builds a massive chemical shield to avoid dying.
Right. And then it activates the mTORC1 pathway.
mTORC1, the master growth switch.
You got it.
Yeah.
The researchers proved this by looking for specific proteins, increased PMTOR, and P4EBP1. Those are the flags that show the pathway is turned on. It signals the cell to aggressively grow, synthesize proteins, and survive.
Which all leads to the ultimate result. The olaparib is trying to cause fatal DNA damage. But this whole glutamine influx, the GSH shield, the mTOR survival signals, it actually restores the cell's ability to repair its DNA.
And they visualize this beautifully using a COMET assay.
Oh, I love the COMET assay. It's such a clever visual trick.
It really is. Basically, it's single-cell gel electrophoresis.
Right. So for the listener, you take the cells, embed them in this agarose gel, break them open, and run an electrical current through the gel.
Right. And if the cell's DNA is healthy and intact, it's a big, heavy molecule. It doesn't move much in the gel. Under the microscope, it just looks like a bright, tight little circle.
But if the olaparib has worked and the DNA is heavily fragmented and shattered,
then those broken little pieces of negatively charged DNA get pulled fast by the electrical current. They streak across the gel.
So it literally looks like a shooting comet with a long tail of shattered DNA behind it.
Exactly. And the results here were so clear. When they knocked down the SLC1A5 transporter, meaning they cut off the glutamine, the olaparib-treated cells had massive comet tails. The DNA was totally shattered.
Because the shield was down.
Right. But when the glutamine docks were open, there were virtually no comet tails. The cell fixed the damage.
And they back that up with other markers too, right? Like gamma H2AX.
Yes, gamma H2AX, which marks double-strand breaks,
Yeah.
and RAD51, which is a protein that physically does the DNA repair. The glutamine metabolism allowed the cell to recruit RAD51 right to the damaged sites.
So the olaparib is effectively useless. The cancer just patches the holes as fast as the drug makes them.
Precisely.
Okay, so what does this all mean for actually fighting the disease? Like, how do we counterattack?
Well, if the cancer is completely reliant on these SLC1A5 loading docks to survive the drug, you blockade the dock.
Cut off the supply chain. How did they do that?
They used a drug called V9302.
V9302.
Yeah. It's an SLC1A5 inhibitor, and it works through competitive inhibition.
So it's not deleting the gene, it's physically jamming the protein.
Right. It's structurally designed to wedge itself right into the binding pocket of the transporter. It physically blocks the glutamine from getting inside.
So they combined this jammer, the V9302, with the olaparib.
Yes. First, they tested it in vitro, so in petri dishes, and the synergy was incredible.
Synergy is a big word in pharmacology. You can't just guess it, right?
No, you have to prove it mathematically. They use the Cho-Talley method. You're trying to calculate the combination index. You want to prove that drug A plus drug B doesn't just equal 2, it equals 3 or 4.
Right.
So a combination index of less than 1 means you have true mathematical synergy. It's not just additive. The drugs are exponentially more devastating together.
Right.
And that's exactly what they saw. Massively increased apoptosis cell death and DNA damage compared to either drug alone.
But I mean, petri dishes are one thing. Proving it in a complex biological system is a whole different ballgame.
Absolutely. Which is why they moved to in vivo models.
Yeah.
Specifically, a PDX model.
Patient-derived xenograft. Let's break that down for the listeners because it's so cool.
It is the gold standard for this kind of research. Instead of using some immortalized artificial lab cells, you take fresh, actual tumor tissue surgically extracted from a human ovarian cancer patient.
And you implant that directly into a mouse.
Yes. An NCG mouse, to be specific.
NCG meaning it's severely immunocompromised, right?
Exactly. It lacks T cells, B cells, and natural killer cells. So the mouse's body won't reject the human tumor.
So you're basically using the mouse as a biological proxy to grow a real human tumor, preserving its actual 3D structure and genetic chaos.
Right. It mimics the real human disease so much better.
Yeah.
And the results were striking. In the mice, just using olaparib or just using V9302 slightly slowed the tumor, but the combination.
Let me guess. Total suppression.
Dramatic suppression.
Yeah.
The tumor shrank significantly. And when they looked at the tissue afterwards, they checked for a protein called Ki-67.
Which is a marker for cell proliferation. It only shows up when cells are actively dividing.
Exactly. And in the combination group, Ki-67 was heavily depleted. The metabolic engine driving the cancer was basically shut off.
Wow. Okay, we're nearing the end, so let's hit our second mini recap before we wrap up. Bullet 1. All that glutamine turns into a GSH shield and restores the cell's ability to fix its DNA. Bullet 2. We can block that loading dock using a specific drug, V9302, which strips away the shield. And bullet 3. Combining that blocker with the original attacker, olaparib, creates this mathematically devastating synergy against the tumor.
It's a very satisfying counterattack.
It really is. So zooming out, what's the big takeaway here?
I think it reinforces this core belief in science that knowledge is most valuable when it's applied.
Right.
By understanding the microscopic why behind the resistance, this crazy USP14 and SLC1A5 dynamic science opened a new door.
Yeah, a resistant tumor isn't invincible, it's just dynamic.
Exactly. We can take the cancer's greatest strength, its hyperactive metabolism, and turn it into a fatal vulnerability.
And that's exactly why this deep dive matters to you listening. It's a perfect example of how zooming in on the smallest mechanistic details, like some molecular scissors, clipping a trash tag off a protein, can solve massive systemic problems.
It really changes how we think about designing combination therapies.
So on that note, I want to leave you, the listener, with a final, slightly provocative thought to mull over, something we didn't really touch on yet.
Oh, lay it on me.
So the study showed that treating the cancer with olaparib actually strengthened the bond between the USP14 bodyguard and the SLC1A5 dock, right? The treatment trained the tumor to lock in this highly specific escape route.
Right, forcing it to become totally dependent on glutamine.
So if our current treatments are actively training tumors to build these predictable escape tunnels, could the future of medicine involve designing drugs not just to kill cancer, but to intentionally hurt it?
Wow, hurting the cancer.
Right. Could we use a first-line drug specifically to force the cancer down a very specific metabolic tunnel straight into a trap where a second drug is already waiting to spring? Like the molecular heist we started with? What if we want them to tunnel out of the vault because we already placed the dynamite in the tunnel?
Evolutionary trapping. Honestly, it is arguably the most exciting frontier in oncology right now. It's a brilliant way to look at it.
Something to think about. Thank you so much for joining us on this exploration of the sources and for taking the time to dive up today. Catch you next time.